Well, thank you for joining us for today's discussion, Alternative Cell Therapies for Oncology, as part of this year's virtual Cell and Gene meeting on the MESA. I'm Martin Lachs, and I'm the Vice President of Project Management for Oncology and Cell Therapeutics at Icon Clinical Research. My own role has given me the opportunity to work across a spectrum of cell therapy clinical trials, going back some years now, so I am delighted to be joined by this illustrious panel whose development programs span a number of technology platforms, which we will talk about. I welcome Stephen Kelly, CEO of Carisma Therapeutics, Filippo Petti, CEO of Celyad, Arthur Lahr, CEO of Kiadis Pharma, and Julian Adams, CEO of Gametocell. Thank you all of you for joining us today. So let me get straight into the discussion, starting by asking the panel to tell us a little bit about themselves and to share with us some of their company's cell therapy development assets. Stephen Kelly.
Sure. Well, first, thanks to ARM for inviting us to participate in the panel. In terms of my background, I've been in biotech and pharma for the last 30 years or so, primarily in oncology, drug development, and commercialization. And I've had roles, running commercial organizations for Amgen, IDEC, and others. But since 2004, I've been doing more startup work and joined Carisma Therapeutics in February of 2018. And it is a unique differentiated cell therapy play. We're working on CAR macrophages, and we're primarily focused on developing CAR macrophages for solid tumor applications. And for a number of reasons, we think that macrophages are ideally suited. They're able to traffic to solid tumors. They're able to kill directly through phagocytosis. They're able to change the tumor microenvironment through cytokine production, and they're able to mount an adaptive immune response. And so those are the things, the hallmarks of our platform. And looking forward to moving it into the clinic and continuing to drive data for patients.
Right, thanks, Stephen. Filippo, what about you and what about Celyad?
Well, thank you again. I'd like to echo Stephen's comments, thanking ARM for allowing us to participate in such a great panel here with regards to cell therapies. A little bit about my background is I'm currently CEO of Cellian Oncology. I joined the company a few years back. My career has taken me through being a bench scientist, working on small molecule tyrosine kinase inhibitors into business development. I spent half my career also on Wall Street, both in equity research and in healthcare investment banking. And so I've had the great pleasure of seeing the industry from multiple perspectives and lenses. I think that's been very useful as I've moved into the operating role at Cellian Oncology the last few years. First as CFO and now CEO. The beauty of taking these different perspectives have really allowed me to strategically think about how we continue to navigate the cell therapy landscape and really put forward our best foot in terms of deriving next generation differentiated cell therapies for the treatment of cancer patients. At Cellian Oncology, I think it really focuses on two really main aspects. We've been really pushing and pioneering biology around NKG2D. This is a receptor that comes out of the natural killer cell, and trying to marry the best of both worlds using this receptor in the context of a T cell effector cell. So we've really pioneered in several programs that we've been working on the last few years, both on the autologous and allogeneic side. In multiple indications. And on the allogeneic side, where we've really been spending time and focusing our efforts of the last few years is in moving towards a non-gene edited approach towards off the shelf. We have two different technologies, a technology we call TIM, and then another technology which uses short hairpin RNA interference. And both of those approaches we really think gives us a differentiated approach as we think about next generation allogeneic CAR Ts and trying to simplify, a very complex modality and putting our thoughts around how do we think about getting the treatments and the available options to patients who are facing unmet medical needs.
Thank you. Well, let's move on to Arthur Lahr. Tell us a little bit about your background and Kiadis.
Yes, Martin, I'm more than pleased to do so, and thanks to Arm for having us here. I spent the first 10 years of my working life in very different fields: engineering, consulting, some other IT. Then 20 years ago, moved into biotech. Spent about half of that at a company called Cell in BD, strategy, M&A, commercial functions. And then 4 years ago, 2017, I became a CEO of Kiadis. Kiadis is an Amsterdam-based cell therapy company. In the beginning, we were essentially a T-cell company with a patient-specific supply chain. Autologous, essentially, running a big phase 3 and preparing for commercial launch in Europe. We learned a ton about all those aspects, getting a cell therapy product onto the market and running large-scale patient-specific programs. But those programs we stopped about 2 years ago, and we switched to the NKSR platform that we had acquired. The NKSR platform is very different. It's allogeneic, off the shelf. It's much more broadly applicable. With the NK cells that we produce, we can see efficacy across different tumor types, and even beyond oncology and infectious disease. It's a very intriguing platform. Essentially, what we do is activate blood-derived NK cells in a certain way that makes them really truly hyperfunctional. With that approach, we can produce enormous amounts of these NK cells. We can cryopreserve them. So it's a perfect platform that is really broadly applicable across tumors, and really can be made available to patients at an attractive cost. That's the core platform. In addition, we're also doing a lot of engineering work with these NK cells. One of those engineering programs, a CD38 knockout, triggered the attention of a partner, Sanofi, with whom we closed a partnership deal about 9 months ago. And that subsequently led to a bid by Sanofi to acquire Chiaris. So very soon we will no longer be an independent biotech company, but become part of Sanofi. And I think we're the first NK cell company attracting that sort of attention of a large pharma.
Thank you. So last but by no means least, Julian Adams, tell us about yourself and Gametocell.
Thank you, Martin, and thank you, fellow panelists. This proves that it will be a really interesting hour to spend together. My background is almost 40 years as a medicinal chemist. I trained in chemistry. I've spent the better part of my career in big pharma as well as startup biotechs, but it's all been small molecules until about 5 or 6 years ago. I had the good fortune to be on the advisory council of Stand Up to Cancer, and I saw Carl June actually present the first CAR T patient, a girl, Emily, who was diagnosed with ALL, and I actually met Emily about 2 years ago. She's off to college today, and so this stimulated my reintroduction to immunology. So I put away my chemistry books, and I still retain some chemistry, of course, but really made a concerted effort to try to understand how we can harness the power of the immune system to fight cancer. And I joined Gametocell about 4 years ago as CEO when I saw the really impressive, in fact, even curative treatments in bone marrow transplant, but also saw the very young program in the NK cell field that we've pioneered for the treatment of lymphoma in combination with rituximab, anti-CD20. And well, we'll talk more about that specifically, but I can really attest to the the power of the innate immune system and immunology in general as a really much more efficient way to target cancer to produce durable disease-free survival. And we've seen that some of our patients are out to beyond 10 years from their bone marrow transplant, but we've seen other patients in the CAR T field, in the whether it's autologous or allogeneic. We're seeing major advances in the field of immunology.
Okay, thank you. So just moving on to the next discussion topic. So what I'd like to be able to is learn a little bit about what you've learned from failures and how that informs you to achieve success in the current cell therapy platforms and technologies that you're developing at the moment. And obviously, I want to hear from you as individuals, but it'd be interesting to sort of share your experiences as we do some crosstalk. So, perhaps starting with Arthur.
Yes, you learn most from failures. That's what they're good for. And with Kiadis, we had our fair share. We ran a large, as I said, we ran a large phase 3 with a patient-specific cell therapy product, which didn't make it. There's a couple of key learnings that came out of this. One is that I used to have follow the general rule that if you have a product that has clinical benefit to patients, there will be a market. And I basically learned that that's not always the case. The patient-specific cell therapy product that we had really had a clinical benefit, but it was much more difficult for the clinics and the practitioners to use because of this patient-specific supply chain than their customarily used post-transplant cyclophosphamide protocol. So we also found this out in interviews with payers and reimbursement agencies, that the logistical challenges would really be a bottleneck for adoption. So the old dogma that if you have something that is better for a patient, it will be used, I don't believe it anymore. You really have to test it all the way through to the healthcare practitioner's ability to actually really use it. The key learning for me from that is that I don't see a future in patient-specific cell therapy. It's just too cumbersome. You can never scale it up. There's certainly going to be niche applications where this is totally fine, but to really get cell therapy broadly adopted, I think the field has to completely shift over to off the shelf. Right. The other key learning is on manufacturing. We've had, if we've ever had trouble, it's always been the manufacturing capacity and quality. If we would have had more money, we would have brought things in-house much more robustly, quickly, and to retain full control over our own destiny. Cell therapy is manufacturing. The product is just completely driven by how it's produced. So I think in that sense, this field is different from antibodies or small molecules, clearly, where you can't just outsource. I think in cell therapy, you want to eventually move it in-house.
So we will probably come back to talk about some of those issues in regards to your current development platforms. But to continue the theme of failures, what we learn from them, I'll move to you, Julian, next. And
Thank you, Martin. So I view failures, there are two types of failures. There are failures that are noble failures, meaning you're testing a theory, you're testing a hypothesis, you're running a well-controlled randomized study, but the theory doesn't match up to the experimental results in a human clinical study. So that's, for me, a noble failure. It's up to us to push the edge of knowledge and test different theories that are supported by good laboratory work, etc. And sometimes our laboratory models just overpredict what we will see in humans. So that's just part of the challenge of science in general. So that's a noble failure. The less than noble failure is when management doesn't recognize what the data are telling them and are pushing forward irrespective of doing of garnering all the relevant information. As Arthur was explaining, if the adopters won't adopt it, and management is ignoring that, that's not a noble failure. That's human error that is intrinsic to the concept of failures. But failure in science is something that we're all very familiar with. We manage failure. That's why it's a self-selecting group of people and individuals who even want to do this kind of work. The rewards are tremendous. It is extremely rewarding to see patients benefiting from a therapy. But it's just a very challenging variable from patient to patient, from donor to donor. In terms of cell therapies, there are many variables that we can't control for, and managing all of that is tremendously challenging and really requires the best and brightest to face the realities that their program might be perfectly sound, but maybe just wrong time, wrong place. Competing programs also come into play, and we can witness this, for example, in the NK field. There are probably 30 companies now that are, if not in the clinic, just about to be in the clinic. So there's a tremendous amount of competitive failure that's part of this discussion as well.
Again, we'll probably touch on a couple of those points, but I'll move across to Stephen.
Yeah, so when you asked the question, I was thinking back on the various things that I've done in the past, and there are so many to really tap into in terms of failures. But I think one really rose to really the forefront in my mind, and has direct parallels to what we're going through with cell therapy. And that was, I was fortunate to be involved in a relatively early stage company, IDEC Pharmaceuticals. We launched and commercialized Rituxan, but then we went to commercialize the first radioimmunotherapy. It was a product called Zebolin. And, I think the things that, it's a good product. I mean, we had activity and clinical activity. There was probably a role for it. But the things that stood out in terms of commercial success were related to, one, product differentiation. We had a product that worked, but we really weren't very good at articulating how it's differentiated from rituximab. And so, I think that we need to learn about positioning and comparing and contrasting the various therapies and what is your role. I think secondly, we had an issue of a logistical barrier to adoption. And so highly complex therapy involving oncologists, hemoncs, nuclear medicine, radiopharmacies. It was incredibly difficult to get the product from us to a radiopharmacy, get it made, get it brought in, various licensing issues. And so I think logistical barriers to adoption are really important in the cell therapy space as well. We have an entire supply chain to manage, and we have to work through that. The last thing I think that stood out were economic barriers to adoption. And so we have a high price therapy relative to the other things that are out there, and not a clear path on who's going to be reimbursing what elements. We have the isotope, we have the antibody, we have the combination with Rituxan. And so working out those economic barriers to adoption and reimbursement are incredibly important. I think that those will remain important for us as we look to continuing to commercialize new and innovative cell therapy approaches. That we have some people that have, or companies that are leading the forefront and establishing those sorts of things, but we have to do those. And so, the failure was, I think, the commercial viability of the first radioimmunotherapy. And, my things that I draw from are clear differentiation, really addressing logistical barriers to adoption, and addressing economic barriers to adoption. Now, those things I think we need to focus on in cell therapy as well.
Yeah, and we will come back and we'll focus that in the specific terms of your own products fairly shortly. But before we do that, Filippo, tell us about your failures and what you've learned from them.
Yeah, look, I think certainly my esteemed panelists have covered many of the topics that we've kind of thought about and faced, and the challenges. It's an up and down industry. Specifically, I think at Celyad, you know, I would say the biggest challenge for us, or one of the failures perhaps, is and how we've overcome that, is the idea of maybe you can't rush, you know, the idea of rushing science. You know, I think we saw some great success in the early part of cell therapies and the CAR T approvals over the last decade or so, and really the momentum that was built there, and thinking about how quickly you could potentially leverage some of that success, and really, you know, as we've positioned ourselves around a novel receptor and NKG2D and trying to prove out that biology. And I think maybe taking, you know, too many broad strokes, and thinking about how broadly we can go with the potential program, maybe rather than focusing our efforts and trying to really prove out the proof of concept and proof of principle around that biology. And it's taken some time. And I believe from our perspective, it has landed us in a great place. But I think sometimes the aspirations and maybe some of the initial data that you see that emerges from the programs doesn't always translate, doesn't always you know, materialize in the way that you perceive it that it potentially could. And I think that has allowed us to, you know, build a larger data set around this receptor and get on an understanding around NKG2D biology, how it dovetails perhaps with T cell biology. And I think also through this process, how quickly, I think, you know, one of our views and perspectives on the industry is how quickly things change, right? I think Julian alluded to this as well earlier, not only keeping an eye on your own programs, but the competitive dynamic in the landscape as well. And I think, you know, resonating certainly with authors that earlier, you know, making this transition from autologous and to allogeneic for us has been a huge endeavor over the last couple of years. And I think we certainly see some of the benefits. And to Stephen's point, thinking about commercial viability around your eventual programs, I mean, it really dovetails to all the panelists' thoughts here. And that has been a challenge for us. And maybe could we have read the tea leaves a little bit sooner and getting us more towards the allogeneic CAR T side of the landscape? You know, we've been fortunate that we've lived on both sides. I can certainly say now that our programs are more geared towards off the shelf. We can certainly point to some of the benefits we've seen. But at the same time, I think it's being patient. This is a novel modality. Certainly, we've seen a few approved products here, but I think we're still in the early stages of development. And I think taking a step back for us as well, it's been how do we get to where the unmet medical need is around solid tumors, and certainly cell therapies have been fantastic for several hematological malignancies. How do we make that bridge? And I think making sure we take the right strategic steps to get there has been certainly an important aspect and reflection for us over the last couple of years.
Yeah, I mean, the investment in biotechnology and cell therapies in particular has blossomed, shall we say, and that gives a lot of potential. But you know, taking into account some of the failures and the learnings that you gained from that, can I ask us to assess critically how good are we actually at applying lessons learned and not repeating the same things over and again? Because, you know, for example, Julian, you said there are 30, you know, about 30 organizations who are looking at NK cell platforms. We know not all of them are going to succeed, but is there an element, and I've seen this a little bit myself, where, you know, we've been doing the same sort of clinical trials of development in assets which are, to use a number of things that don't seem to be clearly differentiated, and it's driven by investment, it's driven by hope. Yeah. But are we good? To what extent? And I throw this question out to you: are we good enough at really objectively learning lessons, looking over our shoulders what the last guys did, and not going down that path?
It's a great point and a challenge for all of us. What we did at Gametocell, which is something was very untraditional in my experience, is we got into the clinic very early in the game. In other words, we had a hypothesis, we had a laboratory method of growing NK cells. We hadn't yet learned how to cryopreserve them. We have since done that. But with fresh NK cells plus rituximab, we could effectuate killing of lymphoma cells in tissue culture and then eventually in mice through a process called ADCC, or antibody-dependent cellular cytotoxicity, knowing full well that the NK cells display CD16 and therefore combine the FC portion of antibodies. So we took a really risky and bold step, but ultimately an economically defensible one, which is we teamed up with Jeff Miller at University of Minnesota and his group, who are pioneering many of the NK cell therapies, collaborating with many sponsors. And we designed a phase 1 study to go with fresh cells into patients with advanced, heavily pretreated lymphomas, particularly having experienced multiple lines of Rituxan-containing regimens. And what we saw in the first 19 patients treated was an extraordinary response rate. We did a dose escalation. We determined that both from a safety perspective and an activity perspective, we have some of the most advanced clinical data, albeit not under our own IND. And so what we've gone back now is refine the process, scale the process. It's an off-the-shelf allo donors are young, healthy adults, which are screened and can provide the substrate for now making the off-the-shelf cryopreserved product. And we hope to file our own IND later this year. What's notable in many of the heme malignancies that sometimes just based on response rates, and we've got response rates in the mid 70% range, both in follicular lymphoma and DLBCL, that suffices as an endpoint for an accelerated approval, followed by a randomized study. So it's a sort of quicker path to the marketplace. But the de-risking element was really to get into the clinic and find out if there's a sufficient signal of activity to continue investing in this program. And I think I won't say we weren't lucky. We were, of course, lucky, but the science was also being well followed from the very beginning. Focus first on safety. That the fact is that we can administer 200 million cells per kilo of NK cells. I think that's perhaps the biggest recorded dose ever administered to patients, and that it was well tolerated and highly active. So that's sort of Gamida's story. And frankly, if we were a bigger company, we probably would go a more traditional path, but it would be a slower, more deliberate path, and not through an investigator-initiated first-in-human study.
So get into the clinic quickly, and that presumably can lead to making quick decisions. I mean, you've been serendipitous, if you can call it that,
Yeah.
Based on good science. But you know, how many of us are killing projects or programs that don't have that fortuitous outcome? So again, it's that lesson learned. I mean, I invite others to sort of chip in on that.
Martin, for me, I love your question. You know, do we, as an industry, learn? It's almost a philosophical question. I mean, I've been in different industries before coming into biotech, and this is by far the most complicated field you can be in. You know, the odds are against you. What's the chance that a drug makes it to market? You need completely different skill sets from preclinical to clinical, CMC, commercial, and the only way to make it is if you work with people, if you build a team of people that have been there and done that. So you avoid the mistakes of the past. It's all about getting the right team around you. And I love the example that Stephen gave about Zebolin. Our chief medical officer, you know, came into the company knowing we had a patient-specific supply chain, and he had been involved in Zebolin himself in the past, and that allowed him to avoid some of the obvious pitfalls with this patient-specific supply chain. And yet, still, of course, there were you know, many of them. And hence I draw the conclusion that you shouldn't move into patient-specific. So without past experience in specific subfields, it's just impossible to get products to the finish. But at the same time, and that's why it's a bit philosophical, if you just always take those learnings, nothing ever gets done. If people would have argued from the start, well, patient specific, let's look at the Zebolin example to just take that. It's logistically too complicated. The products will be too expensive. There will never be an economic model. Then the CAR-T product would never have made it. Carl June would never have pioneered his first CAR-T project and the people working with him. So you, on the one hand, you need that experience. On the other hand, you need to be willing to just believe and push forward regardless of past experience, because that's the only way the field will move forward and completely new things get done.
Yeah,
so difficult.
You take risks, but at the same time, knowing what some of the obstacles or challenges are, there are opportunities to think about doing those differently. So, in terms of managing logistics, for example, which we'll perhaps touch on shortly, but in more detail. But if we're trying to use the same methodologies or the same approaches for logistics management, yeah, sure, we'll come across the same thing. So you build on the experience, but there's still the opportunity to tweak, surely.
Yeah, you have to build on the experience. At the same time, you just have to believe.
Yeah.
And even if past experience tells you, there's always enough reasons to shoot something down. And maybe this is why Big Pharma is not good at coming up with new concepts, because there's always someone somewhere in the organization that will shoot it down. So in biotech, you need to have enough people that know the past and have learned from it, and at the same time are just willing to believe and move forward and solve things on the fly. And some things don't work out, and some things fail, and some biotechs make it, and some biotechs just run against the wall.
Can I underscore? Can I add to Arthur's comment, because it's a really important distinction that he's making, this concept of belief. So belief is a two-edged sword, of course. A champion of a program is considered a favorable trait. Someone who believes in data, someone who interprets data, and then can move forward and make a plan. There's the other kind of belief where the individual driving the program is a zealot, and maybe not paying attention to data, and maybe ignoring certain, lethal aspects. And that's what I was referring to. Management and the teams that you put together, you need to be able to examine your data from a very objective perspective, and you need to pressure test whether your data permit you to continue the program. So I completely agree that we need champions, not zealots. Zealots are dangerous, and champions actually make the sunshine. They pull from different walks of life. They build teams, they build data sets, and they drive to success. And that's critically important. Arthur, thank you for bringing that up, actually.
Yeah, I want to underscore your point. You should never ignore the clinical data or preclinical data. If you ignore data, you put patients at risk. So zealots that do that is just, that's never acceptable. If the data tells you a product doesn't work, you have to drop it, period. So again, there's a balance, but you need champions, because how many products were initially developed for indication 1, they failed in indication 1, and some champion who just believed that there must be something just put it into another indication and the product made it. There's so many of those examples. And without people just pushing regardless of the facts in front of them, other than clinical, proof of, provided there's clinical proof of safety and some efficacy, many drugs would never have made it without those champions.
So let me pull us into the next topic, which goes back to your specific platforms and technologies. And we talked about commercial robustness or possibilities and logistics. So what I would like to hear from you is, where you see your respective technologies fitting into the wider armamentarium of cell therapies. And I'm asking if you can make reference to some of the points, as I say, we've already discussed. Patient market access, manufacturing and logistics, as well as the commercial possibilities. So perhaps I'll ask you to talk about that first, Filippo.
Sure. Thank you, Martin. And look, I think great points Julian and Arthur made as well. And from the perspective of how we're pursuing things at Celyad Oncology, I think dovetails to what the gentlemen were saying. With regards to specifically the data and looking at the data, how do you compare this? One of the things that we've been pushing forward is our shRNA platform. Having the right data set within a landscape, trying to prove out a new technology or platform, I think is important, especially in the early days of cell therapies. So as you can see, the majority of companies within the space are working on either CD19 and BCMA, including ourselves. And a big part of that is because you want to make sure that what you're building, the approach, your methodology is going to have differentiation, because it is a competitive dynamic. And how you push that forward, it might not only be on the clinical data that you generate and how that sits within the other peer set, but what are some of the back end things? And manufacturing for us has certainly been top of mind. How do we make a process that can serve multiple patients off the shelf, but also maybe benefit from an autologous-like process where there's perhaps less manipulation of the cells, the time to grow those cells and produce the lots is a shorter duration to maybe keep the cells healthier. And then I think how that dovetails eventually to commercialization. One of the beauties that we're pushing forward is a non-gene edited approach, and is because we can knock down, let's say, the TCR, at the same time maybe multiplex with the shRNA through RNA interference of not only the TCR complex, but other proteins and genes of interest. And having everything in a single vector, single transduction allows incredible scalability. Specifically, one of the key elements that we include also in our therapies is a positive selection, a selection marker that allows us to enrich for the allogeneic cells that we create. And we do think that as we push forward into the programs, and we're still working on a single donor approach right now, moving to multi-donors, and then thinking about where other effector cells can be leveraged, this all-in-one vector approach, as we like to call it. That should allow us to really think about scalability in commercial prospects. Our goal, in general, I think the goal of everyone who's working on off-the-shelf is to make a cell therapy a modality that's very akin to other biologics, like monoclonal antibodies or perhaps the bispecifics, where treating physicians could easily have access to this and get this to their patients. And as I said earlier, having lived on both sides, we certainly see the benefit of being able to get to, at least in our clinical trials, the cell therapy product to our clinical trial sites and in treatment of patients on a more timely basis. That for us, if we can make that work, if we can get that middle road of having an off-the-shelf product that has maybe a more autologous-like features in terms of efficacy and perhaps safety, that would be a win-win, not only for us, but certainly for the industry and for patients ultimately.
Thank you. I mean, Stephen, would you build on that in terms of?
Yeah, so I was just thinking about how do we fit? Our lead program is a HER2 targeted CAR macrophage. And as you know, there are quite a few approved agents. There's seven antibodies, TKIs, ADCs in front of us. And so how do we integrate a cell therapy approach into a fairly commercially saturated market? And it's a real challenge. And we chose HER2 for a variety of reasons. We think it's an ideal antigen for us to prove our mechanism, our biologic proof of concept. But there's still going to be a need to demonstrate our approach versus other therapeutic approaches. And you think back to your first CAR T is a game changer. You have Chiaris, and so that's really where they were driving, and of course that enables the product to move up earlier. I think that we have to do the same sort of thing. And typical oncology drug development is that you're doing a comparison to existing therapy. And so we're thinking about how do we do a clinical development program that establishes our single agent activity, establishes it in a variety of tumor types and stages of disease, and then how can we integrate with other standards of care? And it's not just, how could we combine with T-cell checkpoint inhibitor or anti-CD47 antibody. But I think we have to go one step further, and is how do we combine with other cell therapy approaches? And I think that we have an immune system, and so there's T cells, NK cells, and all the others, and monocytes and macrophages. And I think that there's probably a way to integrate those as well. And those are sort of some of the things that we're thinking about as we go forward. How do we fit? It's not simply just we have this overall response rate and this extension of survival, but it's how do you integrate and extend the overall patient's life, not just for that one point in time. So those are some of the things that we're thinking about as we go through our development of our platform.
Do you see scale up as being an issue in that context?
I think scale's challenging. I think so. We have an autologous approach, if you look at CAR T, there's some scale issues getting for heme malignancies, addressing the available population. Solid tumors, we're talking about 10 times the size of the market. And so how do we have a personalized approach that goes to the hundreds of thousands of potential patients? And I think the scale is an issue. Absolutely, we're addressing it through enhancing our autologous approach. We'll explore allogeneic, but the big drivers there are reproducibility, vein to vein time, and cost of goods. We are taking an approach that may be unique to monocytes and macrophages, where we can accomplish a one-day process. A closed system one-day process that addresses some of the issues, but certainly the numbers of patients, if you have an autologous approach, there are going to be manufacturing scale issues. Absolutely.
So I'm going to go to Arthur. At the beginning, you were saying that maybe it's a bit hyperbolic of me to say it, but there's a kind of a death knell for the autologous approach Stephen's been talking about, and also Filippo you outlined some of the advantages, perhaps it's too strong a word, of an autologous approach. So when you think about that, Arthur, and then in answer to the question in terms of your own technology, what would you respond?
There will always be a place for autologous approaches, and you always need companies and pioneers that move into new fields, autologous fields, like Stephen does with macrophage. No one has explored macrophages at a large scale here before, and there may be some unique features coming out. So you need that. You need those front runners, and they very well might start in autologous. When I reflect on what we have, our NK cell platform, I think the place for that in the field is on the other side. It's getting a industrial, scalable, robust, economical platform out there that just brings cell therapy to many more patients. That's the beauty of what we have. We have great clinical data. We see remissions in acute myeloid leukemia patients that frankly remind me of the initial CAR T data, with patients that have been completely given up, seeing their tumors disappear. We've had patients with CNS disease that had no prognosis at all, and the brain tumor and brain infections disappearing on treatment with our cells. So that's of course the key. You need to have the clinical data. But what I really like about our platform is that it's very robust. We produce our cells using a very well-defined reagent. Most NK cell platforms use a tumor cell, and with our reagent we can just have a better defined production process, and we have a clear regulatory benefit. We can avoid the use of tumor material in the final production. And again, at small scale with small patient numbers, this may not be an issue, but if you really want to make the product broadly available, you have to meet those tumorigenicity bars and the oncogenicity requirements of FDA. So that's one aspect. The second thing is that we've just seen that we can produce a lot. Out of a single donor, we could produce up to 1 million cells just because we can produce more. We see that it's possible to move into STRs, stirred tank reactors, rather than the more small scale wave bags and systems like that. So with our platform, I think we have a chance to really create an industrial cell therapy platform where we can drive down cost of goods to the level of a monoclonal antibody, and the volumes and the numbers of doses that can be produced suitable for large scale adoption. The other key thing, and I think Filippo mentioned it also, we can engineer ourselves. And the field has a tendency to always use the most sophisticated technical DNA genetic technique because it's interesting scientifically. But in biotech, simple is already complicated enough. So our baseline platform doesn't require any engineering. It's in a way very simple. You take cells from a donor, you mix it with the particle, and you wait 2 weeks. And that gives us, with a lot of underlying know-how on the process development, it gives us lots of cells. All these engineering steps, they're great, and we can do them, they add. But if we can avoid them, we'd rather avoid them because it adds cost, complexity, and again, simple is already complex enough.
Julian.
So I fully agree that ultimately we need to be able to manufacture to feed, to a scale that will reach as many patients as possible, of course. So the allogeneic approach with cryopreservation achieves a lot of that. It's technically challenging in the NK field. We know that NK cells, innate cells, are quite delicate and difficult to freeze thaw. But there are, ways around that. We've figured that out in the lab. The key to our approach has been our platform technology is based on incubating cells with high doses of nicotinamide. And nicotinamide is an allosteric inhibitor of any NAD-dependent process. So what we get is ultimately a lineage of cells that has the right cell surface markers, that still possesses all of the innate biology of NK cells, and actually triggers the adaptive immune response, which is the desired biology of NK cells. That's the normal physiology of NK cells. So I agree with Arthur that over-engineering and over-CARring and over concern about persistence of the cells may be a false lead, actually, in some cases. Let me give you an example. The CD19 CAR NK cells actually wipes out all B cells, not just the malignant B cells, but the healthy B cells. And aplasia for B cells means you can't become vaccinated, which particularly this year is a strong reminder that we can't overdrive the immune system. Otherwise, we delete key cell types in the immune system. So our approach has been kind of a one and done, a single infusion of high doses of NK cells. And for us, a single donor can produce up to 1 billion cells in 2 weeks, based on our cell culture approach, and not have reduction in telomeres. The cells are not exhausted. They do not senesce. We get a high potency cell, which when coupled with rituximab, takes advantage of the biology of NK cells. There's no HLA restriction. We can give huge doses to account for patient-to-patient variability or donor-to-donor variability. And then just the simple application of a well-trodden antibody like rituximab has produced a remarkable high complete response rate that's been quite durable in patients out over 2 years now from their initial treatment. So I think, the field is changing on a daily basis as we learn techniques to both grow cells, freeze cells, optimize their lineage for administering to patients, and being able to run efficient clinical trials that give you quick answers to the fundamental questions. Does your product even work? And do you understand the dosimetry? Do you understand at what dose and over what dosing regimen do you need to achieve optimal activity in patients that are clearly heavily pretreated, which is where we typically begin in phase 1 and phase 2 studies, and go for the nth line of therapy. And all of us face the same macro environment as where do we find these patients. Obviously, they're typically in academic referral centers, and that's the leveling playing field for all of us.
Yeah, it's kind of a couple of things which come out of that. I mean, I think Stephen and also you, Julian, made reference to combinatorial approaches, arguably, and there is a sort of a belief that actually, as we advance cell therapies, we need to be looking at single therapies. There are benefits to that, but to what extent do you see that in reality, that ultimately the success of cell therapies and the various the alternatives available are going to be based on combinatorial approaches with, for example, you mentioned Rituxan, but it could also be with CPIs and.
Yeah, I think, as the toolbox gets more sophisticated, we get more sophisticated as well in terms of designing trials, making sure we can isolate the effect of each component of a combination trial. But, at the end of the day, we are limited by the dose we can give, the safety of the product, whether it's given alone or in combination, and looking at sort of the longer term effects as well, so that we don't find ourselves in a situation where we've wiped out a lineage of cells that's important to normal physiology.
Any other thoughts on that?
I was thinking so it's all about, in this case, it's mechanistic synergy, and in our case we have a macrophage that is an innate immune cell, kills kills through phagocytosis, and it's also an antigen presenting cell, and we're hopefully able to recruit in other immune cells, T cells, NK cells, et cetera, and leverage that normal biology. So on its face value, we could say, all right, well, we have a macrophage. One of its hallmarks is the ability to recruit in T cells. Would a combination of a macrophage plus a CAR T, for example, be synergistic? We don't know that. That's something that we can explore. Or you could say, well, we have a macrophage, and since we are mounting that adaptive immune response, is there some synergy between us and a T cell checkpoint inhibitor? Can we enhance that T cell response? Those are things that we can look at. Other examples would be, we have a macrophage that eats cancer cells. Cancer cells express CD47, it's a don't eat me signal. If you block that, would you enhance the ability to phagocytose? So it's all synergy, I think, is what we're looking at, and it goes back to combination chemotherapy. You have unique mechanisms of action with non-overlapping toxicity. Can we do something similar with cell therapy approaches? And I think that's where we have to drive.
Mm-hmm.
Yeah, I think for us, I mean, we're along those lines as well, especially in our solid tumor approach. One of our programs, our Celyad 101 program, where I think one of the things we've really spent time thinking about the last few years is preconditioning, right? And in the context of solid tumors, our 101 programs in the treatment of metastatic colorectal cancer, where we're leveraging chemotherapy that is typically a standard of care there in FOLFOX and FOLFIRI, and layering on our cells following that as our preconditioning regimen. We've moved to a multiple dosing model for that program. We've seen some very interesting data for, I think one of the leading off-the-shelf approaches for solid tumors. And I think some of that data has, I think resonated with folks in one of our programs, and one of the studies we plan to launch into later this year is evaluating the cells following FOLFIRI, and then eventually layering on Keytruda, pembrolizumab. Can we take a cold tumor using an adaptive cell therapy approach, make it turn it from cold into hot to allow the PD-1 checkpoint inhibitor to really bring additional benefit there, and layering on top of what the adaptive cell therapy has begun to already modulate the tumor microenvironment. And I think that's a little bit around the target, but it is around those approaches, right? How do you make this more of a, again, a biologic, taking it out of maybe the adaptive cell therapy class and just as a subclass of biologics, and thinking about how the treating physicians can really use this in their armamentarium for the treatment of not only solid tumors, but hematological indications.
Okay. So I'll, if there's no more points on that, I think I do want to pick up on one thing that was mentioned earlier about, I think, Julian, you talked about with the limitation in sort of developing in the clinic, because we're going to we're relying on the academic institutions to run clinical trials, and this is, you know, probably my bailiwick to a large extent. Looking forward, how restrictive to the development and the pace of development is this increasing volume of alternative cell therapies, the heterogeneity, the heterogeneity of approaches? How much is that, in your opinion, going to restrict our ability to develop effective cell therapies in a time which is going to support the investment that comes up behind it? When do the investors run out of patience?
It's a really great question. I just happened to look at BioCentury this week and saw that there were 82 distinct agents being developed in lymphoma, in non-Hodgkin lymphoma. So if you can imagine, there are 5 clinical trials chasing a patient in every academic medical center. And how do you choose? How do they triage their patients? How do they decide which patient is appropriate for which trial, and so forth? So that has been a continuous problem in the development of oncology drugs. And it is only becoming more challenging because we have more sponsors, we have more science, we have more product candidates being tested. And so one has to differentiate relatively early, and I would even hazard in phase 1. If you don't see a signal in phase 1, I would suggest strongly that you begin to consider another approach. Because if you don't see your drug or your medicine producing robust clinical data in your first trial out, it's going to be brutally hard to just enroll the trial. And that's just a logistical problem that no company really can manage adequately. Obviously, big pharma can throw more dollars at it. They have more resources, but ultimately the challenge is, to get a minimum number of patients enrolled to produce a unambiguous signal for activity, and then drug development becomes a lot more palatable and achievable.
Any other thoughts on that? Okay. If there aren't, then we're almost at time, sadly. So I'm going to start wrapping things up. And I'd like to thank each of you as discussants today. So thank you, Stephen Kelly, Julian Adams, Arthur Lahr, and Filippo Petti. I think it's been an insightful and fascinating discussion. I'm grateful to all of you, but I also want to thank ARM, the Alliance for Regenerative Medicine, for providing this platform as part of the 2021 virtual Cell and Gene meeting on the MESA, or MESA. We do look forward to meeting in person someday soon when we get through this. But for now, I guess it's me, Martin Lachs. I'm Icon Clinical Research, signing off, and thank you very much, everyone.
Thank you, Martin. Thank you to ARM as well. Yeah. And good luck to my colleagues. I hope all of our approaches work and we have an abundance of riches.
Absolutely.
Same here.
Yeah, all fascinating.
There's no Schadenfreude in biotech. It shouldn't be.